What is...
Hello.
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 The slide is very...
Hello, hi.
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 Hello.
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 People are there.
So we can talk about anything, but supposedly.
Guess so.
Anyone want to start?
I was part of it now.
Sorry, but in fact, it's my first Fireplace session, especially online. Did I miss something, or have you just started?
Oh, so no. So there was. I'm not sure if you saw the present. There was a presentation before.
Oh, I see. Sorry, I missed it then. Okay.
And then there's this fire chat.
Okay.
With no fire, but we're supposed to have a chat.
I see.
So what I presented had to do with metaproteomics and using metaproteomics to understand microbiomes and in terms of function and network, and the fact that using metaproteomics you can get dynamic assessment of functional changes in the microbiome, and that there's a network topology to those microbiomes, and that can change based on insult to the microbiome, like disease or drug effects.
Yeah.
So when we think about you doing microbiome or microbiota studies, that it might be good to not only use metagenomics tools, and I know that sometimes that's the tool that is used because that's what is available, but to go deeper and use other technology to understand, or other ways of understanding deeper the functional changes that are occurring in the microbiome. And as you understand the functional changes, you're getting a little closer to understanding the biological processes.
It was very interesting talk. I was just watching it.
Thanks.
It's the background.
Can I ask something?
Yeah,
I don't know
go ahead.
if you hear me. Yeah, I really enjoyed your presentation, and I'm coming from Finland, and we are studying antimicrobial resistance by using the metagenomics. Now we are, for example, sampling wastewater and some hospital surface samples, the drainage and so on. And we are interested in biofilms, but we have only now done some metagenomics. And as I understood that you have been studying the bowel disease, and if I understood right, you get a quite nice picture of the healthy microbiota, and then you can know what is the disease microbiota. What do you think about using this metaproteomics to study this kind of like very complex environmental samples? Is it possible? And do you know if there's some certain proteins that we know that are in the biofilms? You know, we are kind of like interested to study the AMR in these metagenomic samples, and we would like to know if our samples are in the biofilm form or as a planktonic cells. So what do you think? Is it kind of like some design? And then is it, another question is that, is it always so that you do the metaproteomics so that you look at the proteins and the peptides, not at the RNA and translating that to the cDNA? Because we haven't really been working with the proteins, but with the DNA more. Thank you.
Lots of good questions. So yes, you can use metaproteomics on wastewater and that's been done. There's some labs, and we've done some work here because of COVID related to that. In terms of telling which state they are in, whether they're forming biofilm or not, that's a good question. I'm not a microbiologist, so you would have to ask people for marker of that. But I'm sure that there are some that you can measure. With wastewater, the hardest part is normalization. For example, with people right now, we are measuring COVID-19 in wastewater. That's done using, not by proteomics, but using RNA. They use the presence of standard pathogen, not pathogen, as standard virus in wastewater to measure, to calibrate. And that's the challenge with wastewaters, making sure that your repeat measurement are going to be comparable. But Metaproteomics has been done on those, and we've done some in Canada too, and you can see changes. You see changes based on, in Canada, probably the same where you are, based on winter versus summer.
Okay, thank you. I will.
And your second part was about your question about RNA. You said and versus.
Yeah, that is it always with the metaproteomics that you look at the proteins, not like if you would like to know about the microbes if they are expressing some genes. You know,
yeah,
like
I mean, there's, if you can afford it, there's nothing wrong from doing both, right? Because you're looking at different part of the process. We know that I mean, initially, what people thought is bacteria, when they do two things: they either divide or they produce, and there's proteins respond, and there's a competition between those two processes. But a few years ago, and the thought was you're trying to maximize that. So therefore, there should be pretty strong correlation between RNA and protein if that's the case, right? Because you only produce what is needed at a given time. A few years ago, I don't remember which group, showed experiment in E. coli, and 25% of the measured protein in RNA, 25% of the protein at very low level to no transcript at the RNA. And that's because they were making those proteins and putting them in storage in case of. So through evolution, there seemed to be an advantage to make some proteins ahead of time in case you hit a roadblock and there's no time to make it. So that's, and obviously in the microbiome it's even more complex. So there's a lot of, I think, biological processes. Measuring at different level is fine. You just need to know what it means, though. I think studies that, and we are guilty of the study where we're only doing 16S all the time, is very dangerous in terms of inferring the biology. And people have shown that by 16S, you say there's, and especially people go into wiggle of percentage change, and it's relative. So you guarantee if one change, something else has to change. But when you go into absolute level of those bacteria, sometimes there's no change. So the question is, what is important? Is it the relative abundance change or the absolute level? And at the end, that's really not what is important. What's important is there change in functional output in your microbiome? And if you can put all the elements to get there, then you can maybe better understand. I say maybe because it's a very complex system. So understanding is, even with more data, is not always apparent.
Yeah, and I didn't think about that. The metaproteomics, it's not so expensive than the sequencing. Is it so that you said that if you can afford to do, is it like with doing the metaproteomics and you do it with this mass spectrometry? I don't know.
For us, in my lab, it's cheaper to do metaproteomics than to do transcriptomics.
Okay, yeah.
But if you were to do a fee-for-service place, they probably will see you coming and charge you an arm and a leg. But it's not that expensive.
Yeah.
You know, we spend more time on the bioinformatics than on the data generation.
Yeah. Hey, thank you so much. Now I've been using a lot of your time. I'm sure that there is plenty of more questions. Thank you very much.
You're welcome.
Yeah, Daniel, hi. I also had just a question to follow up on some of the points that you've made. I really enjoyed your talk. One of the points I just wanted to bring up, you just touched upon it, is the relationship between levels of RNA and protein. And I guess there are two major ways whereby one can, of course, explain this. One is the differential molecular half-lives of these molecules. And the other one would be then, of course, regulation. And I think your emphasis on PTMs was absolutely right. But the point is, though, I mean, we need to follow the systems, right, dynamically to really tease this apart. I mean, in future, of course, we're nowhere near teasing this apart yet. And the issue, I guess, also, I mean, I wanted to just hear your thoughts on this in context mainly of the gut microbiome, because the sampling frequency is, of course, nowhere near probably the turnover of the biomass as well as what goes on at the molecular level. So I wondered what your thoughts are on how one could eventually try to tease this apart. I mean, what the relationship is between rapid changes in terms of, for example, a change, when one eats after this, right? I mean, that would be quite different in the morning compared to the afternoon. And how the organisms respond into that in relation to that. And then also, of course, these kind of short-term changes at level of regulation, molecular turnover, and then also in the context of health and disease. I mean, because those are the comparisons that we're drawing, but the dynamics are, of course, very, very different, right? I mean, we're talking about many different timescales here. All right, so there were a number of thoughts. Yeah.
Those are very good points. I mean, we are undersampling in terms of number of samples to do some of those studies. So then you can miss correlation because of that. And that could be part of the reason. When we're looking at PTM, for sure, like what I showed you for lysine acetylation, for example, those have to be the long-lasting ones. So if you're saying very quick response, those we won't see in terms of differentially because we're looking at time scale that are too long. I think that what we need is to move, what we might have to do is move away from the in vivo samples and take those in vivo samples and do in vitro experiment where we can do those tests much faster, and then go back and retest in vivo to see if that's the case. Just because it's like if we say the modification and regulation process that happens in 10, 15, 20 minutes, in human, that would be impossible to sample at that speed by doing colonoscopy and so on. So it might be better to go into a model system where you take that microbiome, put in, and like I showed, 96 well plate, and do those end salt and measure very quickly the changes. And if you see those changes in regulation, then you go back and do those very specific measurement in human or animal model. For sure, the circadian cycle is involved, so that's going to be an impact there. Not just the food intake, because that's not necessarily circadian. And then in other system, at least in human, what people have shown is that you also have diurnal cycles, you have seasonal cycles, and microbiome that's been shown in animals. So if you look at some of the work that's been done on animals like alligators and whales, their skin microbiome has been shown to be cycling seasonally. So I don't know if the gut microbiome, if we had enough samples, if we would see things like that. But all of that is going to be interesting. I think at some point, if you look into what people have done in human, at some point there were molecular biology tools that were put in place that allows teasing that out. So whether we'll have CRISPR-based approaches that will allow us to ask very specific gene-centric questions in the context of a microbiome, if we were to reach that stage, it would be then we can, I think, could go pretty deep into biology.
Yeah. All right. Thanks a lot.
Yeah, you're welcome.
So Daniel, can I ask something? If
yeah, go ahead.
It's okay. By the way, I just read Simon's reply in the chat. Unfortunately, I haven't listened to your keynote lecture. I guess it was today. I didn't know about that. So I was reading about this rapid aim projects that you were doing, I guess. And there are some sentences in that which really is interesting for me. Do you already know what a healthy gut microbiome is my first question. And the second is, if you are using a multiple well plates for this purpose, is it a quantitative test or a qualitative test? The
second part is
is that if you are using a multi-well test for this microbiome assay, is this a qualitative test or quantitative test? Like if you're just like looking for what is inside or how much is inside of which one?
It's quantitative. So you can use as a readout from the Rapidame, whatever you want, 16S, Metaproteomics, transcriptomics, pH. It's you can tie it to whatever readout you want, but it's quantitative. So you can compare across well and across a microbiome.
I see.
And our controls in place.
Sorry, because we were talking about, I'm a parasitologist, and we were always talking about if blastocystis or Giantomoeba were part of a healthy gut microbiota, or were they pathogens. So if you already know if what a healthy microbiome is, in the gut microbiome, did you give answers to that, or?
Oh, no, so that's the first part of your question. So I don't think anybody knows what a healthy, what's the average LT microbiome, and people have been working on that, but there's argument around that, and especially for the gut microbiome, because it might not really be that important, because your gut microbiome was prime basically at birth and educate your immune system. So it doesn't mean that it would just be, then the average microbiome doesn't really matter because you have your own microbiome. There's a paper that came out, was it a couple of days ago, that in animals, I don't remember what they were, either monkeys or another set, they showed that transmission of microbiome is over 97% transmitted. Basically comes from the parents, from the mom. So your microbiome is educated. So we tend, when we do our IBD studies, we do comparison between groups. That's interesting. But the most valuable study is when we follow the same children over time, because then we can follow what are the changes in the microbiome. So then we can follow what happened when they get treated. We can follow what happened when they change their diet. And you can compare between groups, but defining what is a healthy microbiome, I think it's not that easy without taking into account the immune system, and that's what I'm trying to get at. Right? And that component microbiome immune system is far from clear of where that's at. And if you look into infant microbiome when they're born, in the first few days, it goes all over the map. The microbiome changes. Throw in some antibiotics, boom, it changes, and then somehow it recovers. So it's very complicated, but it has to do with the immune system. So I think we shouldn't talk about, at least for the gut, we shouldn't talk about healthy microbiome without talking about host immune system.
Thank you.
Yep.
Hi, Danny. Can I ask one more question? I have a project, we're also collecting the wastewater and also the water from the wastewater and also some environmental sample from a local village in China. And then currently our colleagues have already been, they all analyzed the antibiotics in these samples. And also we are doing some we're also doing some gene sequence, for these water samples, but currently you say the gut microbiota. So I don't know whether, to say, you have already found some links between the antibiotics and also the genes and also the microbiota. I don't know whether which metabolites, we maybe can focus on, except antibiotics and its metabolites.
That's a good question. So in our assay, we can definitely see antibiotic resistance. So if we take a panel of individual microbiome and we screen them against compound, and especially when we screen against antibiotics, we can see antibiotic resistance gene being activated in some of the individual. So, and it's not uncommon. So even in a healthy individual, there's one healthy individual that whenever we take that individual gut microbiome and we treat it with one specific antibiotics, the microbiome grows and antibiotic resistance are through the roof. But it doesn't stay flat, it grows better. But if we treat it with other antibiotics, then it's what you expect, the biomass drops. So you can you can use assays and you can monitor antibiotic resistance genes being expressed. But now your question is whether they are metabolites produced by.
I just to say whether some metabolites, like because currently we analyze the antibiotics and also its metabolisms in the wastewater and other environment samples, but we haven't analyzed other metabolites yet. I mean, the metabolites maybe other endogenous metabolites to say except antibiotics and its metabolism to say whether some I don't know whether like amino acids or some other, or maybe fatty acid to say, or maybe other, to say other short-chain fatty acids, whether they can be abundant in the environment samples, which they are a bit high exposed to antibiotics. I'm not sure.
Yeah, I think it's going to be very complex. Some of them will be present because there's a lot of the things that goes into the wastewater, but also like other than, I would suggest maybe the best is to follow the metabolites of the antibiotics. But if you look just the gut microbiome, and if you look at the metabolomics that's been done on the gut microbiome, there's over 50,000 metabolic features, most of which we don't know what they are. We don't know the structures. We only have a molecular weight based on mass spec. Right? So they could be all sort of compounds. Some of them will correlate with changes in disease state, but we don't know what they are. So we don't know the structure. So then let's say we were able to solve the structure. Then what do? You have a metabolic compound structure. So that's the end product of the metabolic process in the bacteria. How do you go back from that? It's very complicated because enzymes in bacteria are notorious to be able to do more than one compounds. So as long as they have similar features. So then how do you put a whole story in place of how that metabolite, other than doing correlation, how is getting to be important? It's not easy. So you can do those type of studies, but then you can end up opening, like instead of having convergence in your data, you will have divergence. You'll have all these massive things to follow up.
Yeah, sure, definitely. I just say sometimes maybe we have some folks you can focus to say some group of metabolites maybe we can focus on, because otherwise, like you said, because it's like we can say maybe more than 5,000 different kinds of of compounds. You can't trace them to say which compounds from which antibiotics. It sounds very, or maybe from other microbial metabolites. It's very difficult to
I think you have to go back and step back and do in vitro assay like the ones we're doing and ask a specific question. What are the metabolites produced by gut microbiome when you treat with antibiotics A, B, and C? But the problem with antibiotics is you get such a change in biomass that it's very hard to then recalibrate the change in metabolic output. Right? Is it just because everything is dying, or is it really a truly metabolic response?
Yes, cool. Yeah, because it's currently it's in the cohort study. we can't do some experiment to say, okay, this is a baseline, then we add antibiotics, then you have a control, you have another sample with exposed to antibiotics, but here we only, it's a cohort study. We try to collect samples like in winter times and the summer times, just to check the trends or to say the antibiotics exposure levels in that area. Okay.
Yeah, that sounds good to me. I would focus on things that are known and that have logical link. But any other metabolites that you could spend the rest of your life working on those.
Okay, thank you. You're
welcome.
Professor Figys, sorry, just got a couple of questions. Earlier on, I'm a physician trainee in Hong Kong at the moment in infectious diseases. In your keynote lecture earlier on, you mentioned about the resilience of the gut microbiome and how various things can change the resilience. Would you say it's related to, say, the gut microbiome diversity, so that the more resilient it is, the more diverse it is? And
so yeah, I mean that's your starting point, right? So if your microbiome is diverse, more likely that there'll be resilience, because there'll be more than one bacteria able to do the same function.
And so if it's less resilience, then there is increased risk of progressing or causing diseases, such as in IBD patients, that they're more likely, or the chance of having a flare would increase.
So we don't know that they're part of the cause, right? So we just know that in IBD, they have decreased resiliency, but we don't know if this is a cause or an effect of the disease, right? So, but in their state, their microbiome is less diverse, but they are also, when we compare to control, they've lost some of their genius ability to, at least at protein level, do not express specific genes. So that means that their ability to respond would be lower because of that. But that's for their specific state, right? So we don't know how they were before. We would have to do a massive prospective study to be able to do that. I mean, that would be interesting to take thousands of people, follow them over time, and some of them will develop disease, then you might be able to. But otherwise, it doesn't say that. It just says that. But if you have a higher diversity in your microbiome, you should have more redundancy.
And sorry, just one last question. Do you think there's a role in metaproteomics in, say, fecal metatransplant, say, pre-transplant identifying suitable fecal donors, or whether certain sort of feces are more suited to or more safe to proceed to fecal metatransplants?
That's a good question. I'm not sure if there's been a lot of study done yet in that space. That would be definitely interesting to ask the question, because we don't know what is a good donor versus a not so good donor. And well, for C. difficile is one thing, but for applications, if you look at application, and so there's been fecal transplant in inflammatory bowel disease, and there's been mixed results. And so one donor would give good results and another one won't. So that's probably the donor microbiome, there's the recipient microbiome that would be a factor, and potentially a lot of other things. So I think that could be studied in vitro. You could simulate transplant in vitro, like using the assay I just described, and see what's the effect. Because it's not that well understood. Yeah.
Thank you.
You're welcome. Anybody else?
Can I ask something else?
Yeah, go ahead.
Yeah, I didn't get the last part of this fecal transplant issue, but beforehand, when I was reading about fecal transplantation, I was always thinking about how should it be done. Like, for example, do we need to sterilize the person that we're going to do the transplant? Because even if we know that we have a healthy transplant in our hand, the way to use it, like with a patch or with like something to chew or something to what can I say, swallow, should we sterilize the colonic microbiota of the host?
You mean of the recipient?
Yeah, the recipient. Yeah, should we sterilize the recipient, or should we give the transplant just like that? Because there will be a microbiota there. So how should we do that?
Those studies would have to be done, but I mean, for C. difficile, they don't do that; they just transplant, right? But C. difficile is peculiar because it's already taken over. But in IBD, there hasn't been sterilization done or trying to before doing transplant, but there's been kind of mixed results on transplant. I think that you're moving from, I mean, in C. difficile, it's pretty it's logical what's happening because you're basically replenishing a microbiome. In IBD and other diseases, you have a microbiome that is relatively stable. It's not like it's an unstable. So how it's going to respond to another microbiome coming in, and whether you can just sterilize and reintroduce a microbiome, that's a good question. I don't know the answer to that. Yeah. Somebody else had a question.
I have two small questions.
Yeah.
Actually, I find this rapid AM setup is very interesting, and I was wondering, so you use it with compounds to test its impact on the microbiome. Is it possible to use the same setup, but instead of compound, to use a specific pathogen? Because like I was looking into ways to like study pathogen microbiome interaction, like with Salmonella or Campylobacter or stuff like that. And I find the test is very practical, sort of speaking. So have you thought about that, or have you done that before?
Yeah. So we're doing some of that. So we just put a paper on bioarchive, for example, with bacteriophage. We use a commercial bacteriophage that is used for poultry, and we just wanted to see what happened if you treat human microbiome with that. For pathogenic bacteria, the thing is they have to be engrafted in the microbiome. So it could be used. So we had planned. We haven't done that yet. So we have to do tests first. So what happened? So basically, if you had the pathogenic bacteria without engrafting it, the microbiome might just get rid of it.
Mm.
So we might have to treat the individual microbiome with antibiotics first, then add the pathogenic bacteria so that it has a chance to
take out the competition, so to speak.
Yeah, but without taking out the whole competition.
Okay.
Yeah, so that would have to be simulated. We haven't done that with those, but we're doing all sort of compounds. We're doing so we're just about to submit a paper on all the artificial sweeteners and the effect on the microbiome, and they do have effects. The pesticide, because one of the thing in pediatric inflammatory bowel disease is there's definitely a spike in the spring of pediatric IBD patient showing up. And that's usually also when there's more pesticide being used. So we're testing to see if pesticides have an effect on the gut microbiome. So you can just dream whatever compounds you can think of, as long as you have established a condition to it. For pathogen, you would need to make sure that it's representative.
But would, like, so speaking, using bacterial lysate be more suitable or easier to integrate in this setup?
Yeah, you can do bacterial lysate. So we've done that. So we simulated, for example, transplant. What happened if you take the lavage, so from the gut, straight from the gut, from a patient that is a healthy microbiome, and you separate the bacteria and just the content versus the one that has IBD, and what happened if you start replacing in culture one versus the other one, whether it has an effect or not. So you can ask those type of questions, definitely.
And just one last question. From your talk, you mentioned you took 5 donors to take the microbiome samples from to test for the different compounds. And I was intrigued, do all the, like, these donors do the same? Like, would the microbiome have like different impact depending on the one giving it away? Like, I have been reading a lot of studies about, sort of speaking, like genome-wide association studies between the host and the microbiome as specific, and possible human SNPs communicating with specific genes. And I was wondering if you have looked into that with Metaproteomics, if there's host factors or SNPs communicating with specific proteins in the microbiome.
No, so we don't know about specific proteins. What we've done in IBD is we've purified exosomes that are in the lavage, so in the gut lavage. So, and then we've done metaproteomics on the exosome, and we can clearly see changes in the exosome composition between inflammatory bowel disease and control. And then we've mapped so basically, if you do electron microscopy on bacteria isolated from IBD, you will see that they're full of little vesicles attached to them. And when we do metaproteomics on the whole microbiome, we can see a lot of human proteins. They take half of the intensity. The thing that we don't know is we don't know whether those vesicles attached to the bacteria are in fact vesicles that are coming from the host, or they're vesicles that are coming from bacteria, because they also can secrete microvesicles. That we don't know, because you don't see that by electron microscopy. You would have to do labeling and things like that. So, but there are clear communication between the host and the microbiome. They're always continuously pushing against each other. And at some point, there's an imbalance. And that's probably what leads, at least in IBD, to the vicious circle that we see. But when you go into culture, you would not have, you eliminate the host. The only thing we do in culture is we maintain composition and function of the microbiome. It stays nearly identical for 3 days. So for 3 days, plenty of time to do experiment. It's long enough that it simulates the transit time. But you can do that.
Thanks.
I think Ibalas, I think you had a question.
Yeah. Hi.
Did I say your name properly, or this is an abbreviation?
It's Julia. Yeah.
Oh, okay.
So we know that in metagenomics, the DNA extraction kit that we use influences the relative abundances of the species and strains that we detect. Is it the same situation with proteomics? And if it is, how do you choose the method for protein extraction, and how do you validate that so that multiple groups or multiple labs get the same results, so that we can have like a comparable, standardized profile of the human microbiome?
It's a very good question. So we know that just like metagenomics, if you change the lysis condition, you're going to get different results. Depending on what your buffer is during lysis, you might have a whole phylum that are missing. So we've optimized some conditions where we did systematic studies and optimized the condition, and we always use the same condition. But I'm sure we're missing some. There are some bacteria that are extremely hard to lyse. And other than using extremely drastic, the problem is if you use extremely drastic condition, it will impact your recovery at a later stage. So that's the first part of the question. We've published an article a couple of years ago, 3 years ago, saying we need standardization in metaproteomics. Because if you go from one lab to another one, if they use different lysis condition, you will not get the same results. Right? Just like in metagenomics. The standardization is a little harder because you basically need to have some form of a control, either the lysis, or you need to do kind of a bioinformatics standardization, and there might be ways of doing that. Like if you don't, there are high abundance proteins that you have enough, like a little bit like in 16S, you always read the same region, and you're looking for the fact that there's differences in those regions. You can do the same with metaproteomics, just focus on a few genes, proteins, and use that as your readout of how well is it reproducible from one group to another one. That's one thing. In my lab, we also have a protocol where everybody who enters the lab has to go through a pretty extensive training, and they only allowed to graduate from the training once they meet standard of output data. So that's another way we control it. So, and some of them do it right away. Some of them, it takes them a little longer to achieve the same thing. If they don't pass the test, they cannot do experiment.
Seems fair.
Yep. Any other questions or comments?
I have another question. Sorry.
Sorry, you go first.
And I'm sorry if you addressed this in your keynote lecture earlier. Did you manage to get a proteomic profile in different stages of the disease?
So I didn't mention that in this, but yes. So we have patient that we have we followed over time. So we follow them. So we get samples when in IBD, pediatric IBD, when they're newly diagnosed, and then they get treated to induce remission. Then we collect samples every 6 months after. So we have multiple samples from the same patient.
So did you see any differences between different treatments, or did you just aim for mild to moderate and 5-ASA treatment?
No, you see differences. 5-ASA would not be put in remission though. So it's to maintain a remission, right? Usually. So what we see is that if you compare the same individual before and after and compare to a control group, you see that the microbiome before treatment is really far away from control pediatric patient. And then when they get treated, put into remission, the microbiome move towards the control microbiome, but they're not the same. They're still different. So and definitely you can measure that, and you can measure that in different part of the gut. But we've done experiments so that in ulcerative colitis, in some patients, you'll get very clear delineation in the gut when you do the colonoscopy. So on one side, it's inflamed. On the other side, it's not inflamed. So if you go in and you do biopsies right before and at the transition and on the other side, you can clearly see that the expression on the host side and whatever you can do on the microbe from biopsies is different.
Daniel, can I ask a question?
Yep. Yeah, go ahead.
Yeah, I'm wondering just is it necessary to link a Metaproteomics to the detection of antibiotic resistance? If so, what's the advantage of this technology compared the metagenomic sequence or qPCR-based technologies? That's my question.
Yeah, that's a good question. I mean, you don't need to go after antibiotic resistance gene. You can see the expression of them by if they lead to protein expression, that's for sure, and that we detect. I don't think there's any particular advantage than doing qPCR, except that you might be able to detect quite a few more product at once potentially. It's more whether you can assay for it. So in our assay, Rapidem assay, we can assay for those. So we can take one person's microbiome and we can treat it against a panel of compounds. So we can tell which one they are resistant to in straight in vitro assay. But the readout can be whatever you want. It just happened that by Metaproteomics, we can measure it. So when we see it, we report it.
Okay, thank you very much.
Yep. Any other questions?
Can I ask one more question?
Yeah, go for it.
Yeah, just did you compare like the microbiota for AMR patient, for example, like patients with with AMR, with specific like bacteria resistant, and also with the normal people? Did you compare the different?
No, we haven't done an AMR studies. We have, because we do a lot of samples, right? And antibiotics is one of the things that we do in our assay, 96-well plate assay. We've hit healthy adults that are resistant to specific antibiotics, and that we see it right away. But we haven't done AMR microbiome to see what's the resistance. It would be interesting because, well, there's many reasons. You might be able to find secondary compounds that added to antibiotics would still make it work, and other strategies, and so you might be able to develop like polypharmacology for antibiotic resistance. But we haven't done that. But that would be interesting to do. But the readout does, like you could do that in the assay. The readout does not have to be metaproteomic in that instance. Like Chang was asking, you could just use PCR, whatever you want to use, and that would be, I think, that would probably be easier. And because I mean, there are many steps to extract proteins from a microbiome that are very different from going after DNA or RNA. So you can reformat it to be much higher throughput. And don't forget, when we do metaproteomics, we extract proteins. We have to digest them. We have to get them all the way to mass spectrometer. It's quite a bit of step. If you do qPCR, many of those steps, obviously you don't need, so it changes, so you can do much higher throughput. So that would be interesting to do those type of studies.
Thank you.
Yeah, you're welcome. Any other questions? If you're interested, one thing I didn't mention is we now freeze living microbiome. So we have kits that we give to, so we collect samples in inflammatory bowel disease. We collect adult normal sample from the feces, or from pediatric, it's colonoscopy, but most people it's feces. We collect samples in Alzheimer's disease, mild cognitive impair, and normal elderly, anxiety and depression. And we basically have a kit that we give people. They collect the sample at home. They put the sample outside their place, gets couriered to our place, and it's fully anaerobic once they put it into the kit, and it maintains composition and function. So so basically it's about 95% identical to the initial microbiome when you do that, which is pretty good, because the biggest, individual differences is much larger. So we have a kit to do that. So now we collect a lot of, because it's something that people, it's not that easy to get fecal microbiome from people. So we used to have it, and people would have to put it in there. So we say, okay, collect at home. So the parents of kids would be, yeah. And they would stop as soon as you said, yes, but you'll need to put it in your fridge, refrigerator. That was a no-go. So now we have a full kit with ice pack in it and reagent that keeps the stool samples when it's put in under anaerobic condition, so they can ship. Now we freeze those. And we've shown that we can freeze living gut microbiome, and the composition and function remains the same. And we've done that so far, 2-year tests. And I think we should be able to get to 10 years. So that means that that's interesting, because when you get the samples, you can just freeze them and use them when you need them. And you can reprobe them later on for the same individual. Because otherwise it's pretty hard, because you have to get fresh microbiome every time. Not easy from children or from elderly.
I have to ask, because I find this really interesting. Is this like your own made kit, or is it a commercial kit? Or
no, it's our own made kit. So there's a couple of commercial kits. They keep the samples at room temperature. I mean, it's meant for other purpose. The microbiome remains alive in those kits, but they ship it at room temperature up to a few days. And room temperature, the microbiome is going to change. But those kits, commercial kits, the purpose is to isolate bacteria. So it doesn't matter that the relative abundance changes, because you're going to flow sort or whatever and do colonies and all of that. So you don't care. What we care is that we want the composition and the function to remain the same. So if the microbiome composition changes, we cannot use that, because it's not representative of the initial seed microbiome, right? So our kit is designed so that it has a temperature monitoring in it, so that if it changes, we throw it away. We don't use it. We've done quite a bit of tests, shipped from pretty far away to ensure that.
So is it possible to know or to get this kit or to test it?
Yeah, we have that. We have it. We're probably going to. We just have to write a paper on it.
This would be really interesting, because actually, even in clinical settings, we have been doing some metagenomics from clinical samples, and the main thing we see, anaerobics don't survive. Coming from this is actually pretty clear. Like, nothing grow in the plates, and the main finding we see when we sequence is basically anaerobic bacteria. So it would be interesting for more important clinical samples like brain abscess and stuff like that.
Yeah, our clinical kit is pretty big. It's a big styrofoam box to make sure. And we've done tests, because when you get courier companies, they might just park it in a van somewhere overnight. So you have to be able to, because we need to keep at 4 degrees Celsius, so we have to make sure that the kit within 24 to 48 hours, up to 3 days, in fact, will remain the same. Otherwise, you can waste. There's also the reason why your sample would be wasted.
It'd be interesting to read about that for sure.
Can you Can you perhaps put the link to the chat box or how to find this kit?
Oh, so our kit is not published yet, so you won't find
okay.
it. We use it in our publication, but it's not really detailed.
Okay, maybe in the future then.
Yeah, hopefully we just have, well, for sure it's going to be. So as soon as we put everything on bioRxiv because it takes way too long to publish. So as soon as we write an article on that, that will be on bioRxiv.
Sorry, just got two more questions from what you mentioned just now. How much fecal matter do you normally need to collect to run the tests on metaproteomics? And also, what temperature do you keep them under again?
So our kit maintains at 4 degrees Celsius.
So
you don't want
not even minus, it's just normal fridge temperature.
Yeah. Now you don't want to go below 0 because you will start losing bacteria if you do that. Right? Because it's not a freezing kit, it's a kit, it's a shipping kit. Once we get it, we process the microbiome and we freeze them. So we have a different call for that. And that we've worked quite a bit on that. And we're working on new ones with new chemicals to freeze them even longer. Right? Because like, for example, right now we're having COVID issues, and hopefully in 5 years from now, there won't be as many COVID issues. But if you want to do a study on COVID microbiome in 5 years, you won't have access to the samples because they're gone. Right? They're only present when people have it. So if we could freeze them, and we can, then you could come back and do those type of studies later on. So that's our goal, to be able to make those. And there's other microbiomes also that are going to disappear.
And how much fecal matter do you need?
Oh, so that depends. So our tests in our assay in 96 well plate, each well is 1 mil. So and that's we seed the well. So we make a fecal slurry to seed the well, and it's seeded at maximum, I think, 110. So we don't need much. So if we could take 1 normal adult stool samples, once we remove all the debris and everything, we have enough bacteria to do over 2,000 tests.
Wow.
Yeah. And then from that 1 mil vial, you have enough to do metaproteomics, probably metaproteomics and metagenomics, although we usually do 2 vials for that. But the supernatant you can use for metabolomics. So out of 1 mil, you'll get about 0.5 mils of supernatant in the culture, and that you can use, and we do metabolomics on it.
So you are inoculating basically the fecal sample, you are enriching.
Oh, so yeah, so we when we get the fecal samples, the kit back, we have a process. We process it before freezing, right? Because there are things you have to do. There's been paper, at least in genomics, has been shown that if you leave debris in the sample, the genomic results is going to be very different.
Mm-hmm. So how many media did you screen in order to choose the one that you're currently using?
Oh, so that's the paper that we published in Microbiome and Nature Communication. So basically what we did is we use, what's this name? We use an engineering technique to optimize. So if you have the problem with selecting media, so first of all, people were saying you have to have different media for different things. So we selected a media based on 2 things: maintaining composition and function. That's it. Right. And then we set many different variables. And there's an algorithm that you can use. Its name was Japanese, but I always forget its name. And basically, like, let's say if you have 15 variables, it's impossible to optimize 15 variables against each other because the number of tests you would have to do is very large. But there's an algorithm you can put in place to select points that you would test, and that's what we did. You don't end up at knowing exactly the contribution of every single element to it, but you can find an optimum. And so then we found an optimum conditions that maintain composition and function. That's how we selected our medium.
And is this an in-house media or is it a commercial one?
No, but it's a combination of commercial stuff and in-house, but it's published. So if you look into our publications, the recipe should be there. And this, the purpose of that is 2 to 3 days culture. We've done it up to 5 days. At 5 days, you are 85, and in those 96 well plate, you are at 85% similarity to the seed. Reason being is that those are well-based, so they're batch, and you start depleting after a few days. So if you want to go longer, it's possible, but you need to change the format.
I do remember, like previously, when I collected the stool sample for one of my studies, and then I did remember we had a protocol, like I need to, to process the stool sample, the fresh stool sample within 2 hours. But we, in our lab, we do have some, we do put some kit, or put something inside, inside, and then before we process the, the stool sample. I don't know whether in your own kit, do you have that requirement to say you have to process that sample within 2 hours after the participant produced the stool sample, or within
no, so for us
it's a longer
yeah,
time.
it's 3 days after, if it's kept at 4 degrees Celsius. Okay. So and then when we receive it, usually we receive it within 24 hours. 3 days is as far as we've gone, but and yeah, so within 24 hours, usually we get it. That's not an issue. So for us, where we're located, 24 hours means we can ship from anywhere in the US, and prior to COVID, from most of Europe as 24-hour service to North America. Now that's not the case, but we've done up to 3 days after.
That's really cool. It's really, because previously, like when we recruit patients, we only can recruit patients living nearby, or ask patients to come to our unit to produce the stool sample. Because
and that's very hard.
Yeah, it's very hard. That's why you say your protocol can can put that time from to 24 hours, even 3 days, which you can allow more participants to do.
Yeah. So basically, what they do is, as soon as they collect the samples, they put it into our buffer. That keeps it anaerobic, and then we monitor the temperature in the kit. And they have part is we provide a pre-label, so they just call the courier, and the courier comes and picks it up. So for the participant, they can depending on where they live in the building, whatever, they can just leave it in the morning or as soon as they've done, and leave it wherever people pick up parcel, and that's it. That's the only thing they have to do.
That's cool. Thank you.
Yeah, you're welcome. Any other pressing questions? If you tried any of those things and you have any questions, feel free to contact. Basically, whatever we publish, we kind of make it open. So we have people, I mean, I know right now it's COVID, but one day we'll get over it. But we have people coming to get trained. Sometimes it's easier. I mean, it's nice to look at a paper and try to do it, but it's not always that easy. So we have had people coming to just do it, and usually within a couple of weeks, they're able to do it. So that's always a possibility. And for us, the more people do it, the better. Sounds good? Okay, well, great. Nice meeting you all, and wish you a good rest of the conference. And I hope to have a chance to see each other in person one day.
Thank you.
Yeah,
Thank you very much.
Thank you.
Thank you so
very
much.
much.
Bye.
Thank you much, Daniel.
Bye.
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 Bye.